Ladies and gentlemen, single-cell RNA sequencing analysis is firstly introduced in a study focused on transcriptome analysis of mouse blastomas. The study used a modified version of the SMART-Seq2 protocol to perform single-cell RNA sequencing on individual cells. Greetings everyone, and welcome to my channel. Since then, the field has rapidly evolved with the development of various experimental techniques, computational tools, and large-scale projects like Human Cell Atlas, which aims to chart the types and properties of all human cells across all tissues and organs to build a reference map of the healthy human body. Traditional bulk RNA sequencing, which provides average gene expression profiles of a population, unlike single-cell RNA sequencing analysis, provide insight into cellular heterogeneity, rare cell types, and dynamic cellular processes. Single-cell RNA sequencing analysis is a revolutionary technique that allows researchers to study gene expression patterns at the single-cell level. Regarding bulk RNA sequencing analysis, when you take a big bite of the fruit salad and you try to guess the individual fruit flavors based on the overall test, or when you grind the all fruits together, then you might recognize the dominant flavors like apple or orange, but it's challenging to distinguish the flavor or find the elusive cherry hidden amongst the rest. Regarding single-cell RNA sequencing analysis, imagine if you have many cupcakes, each representing a different cell type in a tissue. With single-cell RNA sequencing analysis, you carefully examine each cupcake individually, trying to identify its unique flavor and characteristics. You discover rare cupcakes that taste like unicorn tears and others that make you dance uncontrollably. It's like exploring cupcake wonderland at the single-cell level. Importance of studying gene expression at the single-cell level. Studying gene expression at the single-cell level is crucial because it enables identification and characterization of distinct cell types, rare cell populations, and cellular heterogeneity within the tissue or sample. It helps uncover cellular dynamics such as cell differentiation, cell fate decisions, and response to external stimuli, which are often masked in bulk RNA sequencing analysis. Several packages are commonly used for single-cell analysis. Firstly, with Seurat, a widely used R package for single-cell RNA sequencing analysis, including data pre-processing, clustering, and visualization. Scanpy, a versatile Python package for single-cell analysis, offering similar functionalities as Seurat. Monocle, an R package for single-cell trajectory analysis and pseudo-time ordering of cells. Monocle first employs a differential expression test to reduce the number of genes that applies independent component analysis, then applies independent component analysis for additional dimensionality reduction. scRNA, an R package for normalization and patch effect removal in single-cell RNA sequencing data. In single-cell RNA sequencing analysis, there are several unique expressions and terminologies specific to that field. Here are some examples. Cell clustering. In single-cell RNA sequencing analysis, cells are often clustered based on their gene expression profiles to identify distinct cell populations or cell types. Terms such as cell clustering, cell analysis, or cluster identification are commonly used. Dimensionality reduction. Single-cell RNA sequencing analysis generates high-dimensional gene expression data, which can be challenging to visualize and analyze directly. Dimensionality reduction techniques like principal component analysis, T-distributed stochastic neighbor embedding, or TISNE, or uniform manifold approximation and projection are frequently employed to visualize the data in lower dimensional spaces while preserving its structure. In addition, pseudo-time. Pseudo-time refers to a continuous ordering of individual cells along a trajectory representing biological processes such as cell differentiation or development. Pseudo-time analysis interferes the progression of cells through computational algorithms, providing insights to dynamic processes at the single-cell level. For example, imagine you have a big puzzle with many pieces and you want to put them together in the right order to see the complete picture. In single-cell RNA sequencing analysis, scientists have a special kind of puzzle, but instead of puzzle pieces, they have the cells. Each cell is like a little piece of the puzzle, and they want to understand how these cells change and grow over time. To do that, scientists look at the cell's genes, which are like instructions inside each cell telling it what to do. They use a special machine to read and measure the genes in each cell. This gives them a lot of information about what each cell is doing and how it's different from other cells. Trajectory analysis involves studying the developmental path or trajectories of cells over time or under specific conditions. It helps understand the progression of cell states and cell fate decisions in cellular processes. Imagine you are playing a video game where you have a character going on an adventure. As your character moves through the game, they encounter different challenges, collect items, and progress different levels. In single-cell RNA sequencing analysis, scientists are also on an exciting adventure, but instead of a video game, they explore the cells inside our bodies. There are many different types of cells, like tiny superheroes with unique powers. Scientists want to understand how these cells change and interact with each other during important processes, like growth or healing. This is where trajectory analysis comes in. Trajectory analysis like creating a map of the adventures the cells go on. It helps scientists figure out how different cells change and move from one to another. Cell type annotation. In single-cell RNA sequencing, identifying and annotating cell types is a crucial step. Cell types annotation involves assigning or labeling cell clusters with known cell types based on the expression of specific marker genes or reference databases. Imagine you have a big box of colored pencils and you want to sort them into different groups based on their colors. Each color pencil is like a cell in our bodies, and scientists want to figure out what type of cell each one is. In single-cell RNA sequencing analysis, scientists use a special machine to look at the genes inside each cell. Genes are like little instructions that tell each cell what to do and how to behave. By reading the genes in many different cells, scientists can start to see patterns and understand the different types of cells that make up our bodies. Cell type annotation is like giving a name or label to each group of cells based on their similarities. UMI counting. Unique molecular identifiers are short random sequences added to the individual RNA molecules during library preparation in single-cell RNA sequencing analysis. UMIs enable the quantification of RNA molecules and help distinguish true biological molecules from PCR duplicates, improving the accuracy of gene expression quantification. Imagine you have a big jar filled with colorful messages. Each message is like a special secret code. Scientists want to study these messages and find out how many times each code appears. They do this by using unique molecular identifiers, or UMIs. UMIs are like special tags attached to each message that help scientists count them accurately. These tags are special stickers with unique numbers on them. Each sticker is different, just like each UMI is unique. When scientists study the messages, they use a special machine to read the codes on the stickers. This machine tells them how many times each code appears in the jar. It's like counting how many times each UMI shows up. By counting the UMIs, the scientists can figure out how many messages have the same secret code. It's like counting how many red messages, blue messages, or green messages are in the jar. The UMIs help scientists keep track of each code and make sure they count everything accurately. UMI counting is important because it helps scientists understand how active certain genes are in different cells. Genes are like special instructions that tell our cells what to do. By counting UMIs, scientists can see which genes are more active or less active in different cells. Imagine if the messages were cells and the secret codes were the genes inside the cells. By counting UMIs, scientists can learn exciting things about how our bodies work and how cells are different from one another. These expressions and terminologies reflect the specific challenges, methodologies, and analysis involved single-cell RNA sequencing, distinguishing it from bulk RNA sequencing approaches. Thanks for watching. I hope you enjoyed the video. If yes, then please don't forget to subscribe to our channel, hit the like button, share the video, and leave your comment under the comment section. Until next time, happy analyzing.